We have Dileep George, who's going to give us an official workshop presentation. We'll get a sneak preview of next week. Please.
Great. Thank you. It's great to be here, and it's great to give the presentation 1 week earlier. So I'm not able to give all the things I promised, but some of the things it will take another week or so. So the talk is going to be mostly about hippocampus and a very different way to think about hippocampus compared to what you might have been used to. The summary would be that space is a sequence and hippocampus is just doing sequence learning. And most of the phenomena that we see in the hippocampus can be explained just using the sequence learning idea. And I'll present a very concrete model for that. And I will connect it to what prefrontal cortex is doing and the idea of schemas, and also connect it to in-context learning and transformers. And then work towards a system and microcircuit level model of the hippocampus. It's analogous to what we have done in for a model of the visual cortex. Using the same mechanism, I will argue that we can derive a system level model for the hippocampus, which I haven't done yet, but I have a sketch of it. Okay, so if you work in the hippocampus, you might know a lot of phenomena. There are place cells that you all know about, but there are also boundary vector cells, landmark vector cells, splitter cells, sequential order cells, lab cells, place cell repetition. There are various kinds of remapping. In fact, if you do an experiment in the hippocampus tomorrow, you will discover a new phenomenon that you can write a paper about. But my claim is that all these phenomena that look different are really all the same, and they can be explained using our model. And it's based on this paper. Most of the talk is based on this paper. Now it is in press. It's accepted in Science Advances and hopefully be out soon. Okay, so let me present what the problem an animal faces is when it encounters an environment. So here is an environment, a T-maze. Animal is in that environment and it sees just an image and maybe other sensations, but just focus on image for now. It just sees a sequence of sensory observations and it does not have a GPS. It does not have a compass. So all it gets is a sequence of visual sensations, and it has to figure out the layout of the room. It has to create an idea of create a cognitive map of this room or this maze it is in. And the trouble is that the sensations do not tell directly about locations. It's not like a one-to-one mapping between sensations and locations. You can get the same sensations in different places in the environment. You might by introspection say, oh, that's not true. Every place looks different to me. No, that's because you are a learned system and you are utilizing all the machinery to disambiguate. But if you just look at raw sensory observations and try to even do a cluster analysis of them, you'll see that the same image occurs in multiple locations. It's called perceptual aliasing. It's not a wildly surprising thing. It's perceptual aliasing. Another way of saying that is world is partially observed. World is not fully observed. We are living in partially observable environments, and you need to learn the structure of the environment from partial observability. And here is a more understandable caricature of the situation. All the results that I'm going to present are in the 3D environment, simulated 3D environments. But this is just to understand the situation even more. Imagine the rat is in a 2D maze like this, and the walls are green color, orange color, etc. But the carpet is gray, and the rat can see only locally. It only has local sensations, and it is moving around in that room, foraging in that room. And we are not making any Euclidean assumptions. We don't know how the space is organized. It's only a sequence of sensations coming in. So that means, the sequence of sensations the rat sees as it walks is gray, gray, gray for a while, then green, green, green for a while. And of course, it has motor actions along with it. From that, it has to learn the structure of this environment. And if you apply our model, which is called CSCG, clone structured cognitive graphs, it will actually recover the layout of the environment. So just the only thing going in is just the sequence of sensations and the motor actions. The motor actions are kind of optional. It doesn't, it just recovers the graph a little bit better if the motor actions are there. If not, it will still recover a graph. This map has a one-to-one correspondence with this environment, and this is non-trivial. It's not. If you just do sequence learning in the observed space and do not create a latent space, this doesn't happen. And also, this is independent of the topology of the environment. The rat can be running on the surface of a ball. It will recover the structure. On a Rubik's cube, it will still recover the structure. The same problem, aliasing, happens there too. So it's a partially observable environment, and it's recovering the structure. And the environment doesn't have to be Euclidean at all. The environment can be non-Euclidean, and it will still recover the structure. Now let me, and so here, what you've seen is that without using any Euclidean assumption and just using sequential information, it is possible to get the layout of the environment. It's possible to recover the structure of the environment. Keep in mind that the structure of the environment is just a graph. I am plotting it in 2D or in 3D just to visualize. The animal doesn't have any notion of that, 2D or 3D-ness in it. It's just a graph. And all the operations are just operations on this graph. And space is just an emergent property from operating on this graph. So let me just, give you the mechanics of how this representation is and how the model works. So this is the space that we are in. This is the this is the space we are in, and you can see that there are only 9 different observations in that space, 9 unique observations. The number of, if I just grid it into different locations, there are, I don't know how many locations, let's say 100 locations, maybe 81 locations in it, but there are only 9 unique observations.
Where are the 9?
The color. The color, imagine that the color is the observation. Okay, the local sensation is the color. So there are 9 unique observations.
It's the color of the sector.
Yeah,
not what you're looking at.
That's correct. The color where you are in, it doesn't matter. It can be one step ahead, doesn't matter. Yeah. And this is just a caricature. In reality, it is looking at an image which maps to an, after vector quantization would map to a number. I'm just representing that number as just a color. Right? So think of it as the input is one vector where there is one location, it's a one-hot encoding of the observation space, and whichever color is currently active is that's the one which will be active. Okay, that's the input. So there are 9 unique observations. If the world is continuous, you just quantize it, and this, instead of 9 different observations here, you might have 1,000 different cluster centers, but the representation doesn't change. The model or representation doesn't change. Now, so that's the observation space, and there's the latent space where we create clones of these observations. Here is a blue observation. All these internal latent states are clones of them, in the sense that they are really wired to that observation bottom up, but they have transition matrix between this latent space where they will make different connections and they will differentiate. So bottom up, from the observation point of view, they are all clones of that because they are all connected to the same observation, but their temporal context will change because they will connect differently to different clones in the latent space. Hopefully, it will become clear in the next few steps. And so when we start the learning, so as I mentioned, input at every time instant is just a one-hot encoded vector of which observation that you're seeing. And when we start learning, each observation is hardwired to all these clones, and that hardwiring doesn't change. And initially, the connections between these clones are random, and that's what we'll adapt with learning. Okay, so imagine that all of them are randomly connected to each other. Very hard to show in the graph. And after learning, in this particular case, after learning, it will they will get connected in this form. Okay, this is from the random connections. All we are doing is the learning update that I will mention later what the update mechanism is. It will learn this set of connections. This is the graph it learns, and this is the same graph that I showed you earlier. It just all I'm doing is I'm taking this graph, removing the unused clones. So it's just the clones that are connected, and then if I change how it is laid out, that graph corresponds to this graph. This was the graph that I shown you earlier. So just to show you that this is how the graph is organized, and I'm just taking that graph and changing the layout, but the connectivity is exactly the same. If you look at this left image, here is the gray neuron, which activates all these neurons bottom up. These are all the gray clones bottom up, but they have, you can see this connectivity between them, which corresponds to the layout of the room that it has learned. Okay, so now we can ask the question. Okay, so this, I've changed things slightly, but hopefully you won't get confused. This has 4 copies of each of them, because this was, this experiment was run in the egocentric setting where the actions are with respect to the agent. So agent, basically the head direction is an inferred. In addition to location, the head direction is also a latent variable that is inferred. So there are 4 head directions in every location. That's why there are four clones corresponding to each location. So that's also something that the model learns. You're not inputting it in the model. Okay. Now we can ask the question, what observation does this clone respond to? This, you can think of each of these states as a neuron, so I will use the word clone, state, neuron, etc. interchangeably, and we can ask the question: what observation does this neuron respond to? What observation does this respond to?
You said 4.
Yeah, but what's the sensation it responds to?
Not just vision.
Yeah, what is the sensation? It's just response to gray color, right? It's just response to gray color, right? That's what the remember, if I go back to the previous one, all those neurons are wired to this observation, so they respond to gray color, correct? They respond to this neuron responds to gray color. What do this one respond to? That also responds to gray color, right? Both of them respond to gray color. That's they're hardwired to respond to gray color, but they respond to gray color only in specific contexts. They don't respond to gray color everywhere. They respond to gray color only in the specific sequential context. So if I'm talking about this particular neuron, that responds to gray color when the gray color observes in this particular sequence. Any of these movement sequences, will result in that neuron firing. Does that make sense?
What are the different arrows there?
This is where if the animal is moving like this and reaches here, that neuron will respond. The animal is moving like this and reaches here, then neuron will respond. Does that make sense intuitively? Yeah.
So in other words, it's not just first order Markov. It has to be the entire.
Exactly. Yeah. It's in, because this graph is in the latent space. It's not first order Markov. It's first order Markov in the latent space, but it's higher order Markov in the observation space. Yeah. Does that make sense?
Yeah. Well, it just seems like that the number of ways you could possibly get there would be so many that you could possibly.
Oh yeah, but as long as you have inferred the correct latent space, as long as your latent graph corresponds to the topology of the environment that you are in, all the different paths are encoded in that. So this just naturally happens due to how the graph is structured in the latent space. So all these, yeah, there are all these different graphs, all these different sequences that can lead to that location, but all of them are represented efficiently in this graph because it has input the latent structure of the environment.
What if there's no trajectory and you just drop in one of those gray spots?
Oh, I'll come to that. Okay, I'll come to that. Yeah.
What prevents it from just learning like a 1D line of like the sequences itself?
Oh, if it experienced, so this is learned, this graph is learned using foraging. It's doing a random walk in the environment. Of course, it can't see all the potential sequences, but it has to see an adequate number of sequences to infer the structure of the environment. If the agent experiences the world only as a strict set of sequences, if the agent experiences the world only by going from left to right, then it will only have that in the model, and its model will not have this particular property. In fact, the neurons will be directional sensitive at that point. Okay, so now if I plot the place field of this neuron, well, you know how we plot the place field, right? We impose our constraints onto the animal. We basically say, oh, I did the experiment in this rectangular room, so I'm going to put a rectangle there, and I'm just going to plot. I'm just going to put a point whenever this neuron becomes active, right? That's how you plot the place field. And if you plot the place field of this neuron, it will look like this. That makes sense, right? And it will look like the neuron is responding to that particular location. But it's really not. It's really responding to the sensation occurring in a particular sequential context. This idea that it is responding to this location is an artifact of our experimental protocol. And this will become obvious when I show you some of the manipulations I can do. So the place field of this neuron would be this, the place field of this neuron would be this. But keep in mind that the animal doesn't think about the geometry of the room or anything like that. There is no width in this graph. There is no height in this graph, right? There are no walls in this graph. It's just a graph with a bunch of connectivity, and there is no geometry in it, right? And it's just a connection between observations and latent states and connectivity in the latent states. And what does this graph correspond to? If you think of it in an abstract, it's just a compression of its episodic experience. It's just a compressed latent state representation of episodic experience. And note that location is not an input to the model at all. This is very different from other hippocampus models, where most of the hippocampus models start with putting location as the input to the model, which is doesn't make any sense, because location, if you input the location, there is nothing to be solved really. Yeah. Okay.
So when you do your random walk, you get this line graph where everything is in degree 1, out degree 1 of observations, right? So then there was this compression step where you got to a graph that has, doesn't have this in degree restriction. How does, are you going to talk about that, or how does that happen?
Yeah, there are multiple ways I can explain this. So input is not a graph, right? Input just is a sequence.
I know, but it's like, you can think of it as this directed transition graph. It's just a line.
Well, sure, yeah.
Like, I agree that this graph represents the environment.
Yeah.
But the hard part is getting from this just sequence of observations, and then you infer that some of these observations are the same.
Yeah. Okay. So let me try to intuitively explain it, and then the mathematical explanation will be it might not give you any intuition. Okay. So let me try to intuitively explain this. Okay. So when the first, let's say we are just starting out in the environment. When the first gray, let's say the first observation is gray that we get. Okay. Suppose we just select, since we are just starting out, we select one of these neurons randomly to represent that particular observation. And then let's say we just try to memorize this whole sequence as it is happening. So by picking a unique neuron every time, you will be able to memorize the whole sequence. Does that make sense? If the next observation is also gray, I connect one gray to the other gray, and you can memorize the whole thing until I run out of capacity. Agree? Now the question is, what happens when I run out of capacity and when I come back and say, oh, I am at the gray neuron. When I want to connect to the green neuron, I run out of capacity. Okay. Then which neuron do you connect to when you run out of capacity, right? The answer is you connect to the neuron which is going to predict the next observation. Does that make sense? Because all of them are out of capacity. So which the least detrimental one would be the one which would actually is going to predict your future. So if you look into the future, you can see that, oh, I will pick the neuron to connect with the one.
But
And
I mean, don't you have to look like quite far into the future to know
yes,
that
yes,
the work
yes. Yeah. So you have to look into the future to, but we have experiments show that even looking 1 or 2 steps ahead is good enough. So this is the mechanism, which is basically saying, oh, by doing this in a smooth way, I'm describing the intuition, while doing it in a greedy way.
Yeah.
Whereas by doing this in a smooth way, where you are basically looking at, oh, what are the right connections to make in the latent space, you effectively learn the graph. That's the, yeah. And this happens via expectation maximization algorithm in a very smooth way.
Like you plug it in maybe.
No,
Sometimes.
sorry, why is that?
Not advancing.
No, the projector.
Blurs it out.
No, they just.
Oh, I didn't realize.
So one question.
Yeah.
Well, monitor is down. Can you make the life of your model too hard? Because I mean, there are proliferating cortex or these inputs about location.
How does it get that? How does the entorhinal cortex magically know about location?
So in a way, you're solving the problem, which is way more difficult than what you would need to solve.
No, but this is just shifting the problem elsewhere, right? Entorhinal cortex does not have a GPS.
No, but there is all this location representation.
But how though? How?
All the assumptions that was.
You think of it as doing path integration.
Yes. So it can have short distance relative locations. So that will help the model, but that doesn't help you form long distance maps because path integration drifts, and it can give you short term mostly like, okay, I am at a different place from the previous one, and there's some short distance vector integration, but it doesn't give you anything more than that. And that, if you feed it into the model, that will help the model learn the representations especially in the uniform sensation area where there is no distinct sensations. It just helps the model learn better. So if you, basically this is saying, if the grid cells are there, which is from the path integration, it is an additional signal to the model to learn. But keep in mind that if you have 2 different locations that are aliased, and the grid cells are not going to be able to distinguish between them. Grid cells are just going to tell you that, oh, relatively in that each of those locations, this is your movement, but you won't be able to form. Suppose there are 2 identical rooms in the building, and they are separated by a distinguishing corridor. The grid cells are not going to tell you that, oh, these 2 rooms are 2 separate rooms, and they are in a building in a global context.
There could be other contexts that could tell you that in the other parts of entorhinal cortex, besides the grid cell that might have.
But what can they have? It has to be sensory information, nothing else. Because there is no location is never fed into the model directly. Location has to be an inferred thing, a learned latent thing.
But do you, I guess in your model,
maybe you're getting to this later in the talk, but I'm wondering, because basic cells, they emerge like relatively quickly in your environment, and I'm wondering,
Yes. That's definitely, as expressed, that's a deficiency of the model, that it requires quite an extensive experience. If you start from scratch to learn the place cells, I will address it when I come to schemas. Okay.
Thank you.
Okay, so now let's do some experiments. Let's replicate a. Okay, so as I mentioned, we can plot the place field of a neuron by basically saying, hey, where does the neuron become active, by putting dots in the places where the neuron become active, right? That's how we plot the place field. And the place field of this neuron is this. Now we can ask the question, and that neuron is this, that's it, one-to-one corresponds. And this is all things I explained. So now we can ask the question, if this is the place field of the neuron in this environment that I've been trained on, what will happen to the place field of this neuron if I elongate the environment during test time? Okay, so I train the rat in an environment of 9x9 or whatever 10x10 size. Now I just elongate the environment during test time. I want you to predict what will happen to the place field of this neuron. You will be, you'll be running a experiment that was done by a Nobel Prize winner. So you have to. So don't let the layout of this graph fool you. Keep in mind that we are asking you to predict the place field of this neuron when the room changes. Okay, the room change doesn't correspond to a graph change. Only the environment changes. See, the rat doesn't even know that the environment changes. Whatever change it detects has to be through this graph, right? It has no other mechanism, right? So let's run this experiment, okay? What we do is we take this original room, we elongate it, we just stretch it out. Keep in mind that nothing really changed in terms of the number of observations. It still has the 9 unique observations. We just extended the room, right? In some other models, which directly sense the locations. Oh, now there are more observations. No, here the same number of observations, 9 unique observations.
I don't understand what elongating the room, how you do that or what that means.
Oh, this in the experimental setting, the rat
is in a box.
In a box, yeah.
In a bigger box.
Yeah, that's right. Yeah, all the other sensations remain the same. The walls colors are just extended. Yeah, so that's what's done. Okay, now we then do what experimenters do. We let the rat run in that room, and we plot the place field, which is mark the locations where it will become active, and you will see that the place field will have a bimodal representation. Now it will split it into two peaks, and with a soft center in the middle. This one you'll be able to run this in your head, but I have a simulation of this in slides. I will show you next. So this is a famous experiment by John O'Keefe, and it is typically explained as remapping with respect to boundary. And there is a model called boundary vector cells, which is saying hippocampus represents boundaries, and the vectors from the boundaries, all of them to in a very very geometric terms. But here it just happened without anything, just running our sequence model. So now let's see how this happens. Remember the rats. This is the graph inside the rat's head. Although you elongated the room, the graph in the rat's brain is still this one, and we are going to see what happens in the rat's brain when it takes this path through the environment. Okay. So when it is at location A, so I just picked up the rat and put it in A. What cells will be active in the rat's brain?
This is the first time.
First time, but the rat has seen this room before when it was not elongated.
No.
Normally.
Now you're
Yeah.
seeing it first time.
Yeah. So when I first I put it in the gray location marked A, which neurons will be active in the rat's brain? Yeah, all the gray neurons will be active because the rat doesn't know what is the context. All of them will be active. Makes sense, right? Okay, because all of them are supposed to respond to the gray sensation when there is no context. Now let's say rat took this path. It turned over, left, right, all these things, and now it's at B. Okay, which neurons will respond? All of the green neurons will respond at this point. Does that make sense? Okay. Now the rat goes up, turns left, and then goes right. Okay. Oops, sorry. Okay. So now it's at C. Which neurons will respond? actually only one neuron will respond at this point, not all of them, because from the green, if you take one left and one right and reach pink, there can only be one location, right? And keep in mind that this is all probabilistic. This doesn't need to be like exactly like this. If the input is uncertain, it will do the correct computations to reflect the uncertainty. But at this point, even though it hasn't seen, it didn't have to go to a unique place like yellow, which is actually unique. No, it didn't have to go to that. At this point, the rat knows where it is, now let's like
Because of the previous environment.
What?
It knows where it is because of previous
yeah,
environment.
previously learned, because this is the graph it is operating in. This is from the previous experience.
So initially it didn't know where in the blue it is, but
Now it thinks it is in this room in this location. That's what the rat thinks.
So the thing you said about no context, I mean, it's such a reply if we didn't do it as well. Like if we just put the rat back in its old room with no context.
Yeah, this is exactly what will happen.
The same thing would happen.
Yes,
right?
this is yeah. Now we will come to the so far everything that has happened is it will be the same in the old room and the new room, but things will change at this point. Okay, so let's now rat comes to D. When the rat comes to D, this neuron will be active because it thinks it's in the old room. And when rat comes to E, this neuron will be active, and this is the neuron we are recording from. Okay. This is we are asked to plot the place field of this neuron. So when it's rat is at E, that neuron will fire. We'll put a dot there. Okay. Now what happens? The rat keeps going forward. When it's at F, what will happen?
now it knows it's not.
Yeah. So when it's at F, it was expecting to see blue, but what it is is gray. Okay. Now, depending on the smoothing setting in your model, like there are different ways to handle it, but it's like this is a surprising thing. And what usually happens is you just crank up your uncertainty and basically say, oh, I will think that I made a mistake somewhere. It's a surprising observation happens. Probably what happens in the probabilistic model is that it now discounts the observation. And it latches on to a previous model, and it basically says, oh, I am now uncertain about where I am. So the uncertainty but is restricted to these gray states. Yeah.
So just clarifying the question, when the rat is traversing and gets to like F, for example, right?
Yeah.
You said before when we dropped it in A that all of the gray neurons are firing because that's what it's experiencing. When it gets to F, but are the different border colors firing in addition or just those?
Just the one. Again, all the firing is like what is the firing at this instant, right? So when it gets to F, only that neuron is firing. The other neurons did fire in the previous time step. The border neurons did fire in the previous time step. But that context is then carried over in the latent state. Not yeah. Okay, so now the rat, when it reaches F, when it reaches F, I think all these neurons will fire, and so at this point the rat is uncertain about which of these gray states it is in, and it will keep being uncertain about it for a while because it is constantly expecting to see either blue or one of these gray states. But until it reaches the blue state, it will kind of remain uncertain. So it will have this low activations during this period. And then, so that's all the way here. We will be putting small dots in because this neuron that we are recording from will keep firing a little bit because of this uncertainty. And when it reaches the blue thing and it finally observes the blue, it's a strong observation now, and it kind of resets its context. Then when the rat walks back, so now it has seen the blue, and now it is walking back and reaches this position H, our neuron will fire again. Does that make sense? So this will happen over and over again. And that's what creates the bimodal receptive field. So no geometry concept invoked at all. Just falls out of just uncertainty and probabilistic inferences. Yeah.
Can you go back to when the circles at F?
Yeah.
Okay,
Yeah.
why would it be that uncertainty is only horizontal?
Yeah.
Why would it be vertical?
Yeah. So, oh, I see. Why wouldn't it be vertical? Because it knows that its horizontal position, its position vertically is kind of it's more certain about that. Right. It's none of this is, I'm not. It's more like, imagine the input had noise in it. Which direction does the noise spread? That's the way to think about it. And it will spread more in the direction. Oh, I see. Oh, because the motor actions are in this direction only. In the direction in which it is going. Does that make sense? If there was no motor observation, it will spread vertically and horizontally equally. But the motor actions, like the motor commands, tell you which direction you are going.
So there is the presumption that noise, as if it moves horizontally, the noise will also only be horizontal.
Yeah, because it is spread. The computation is conditioned on the motor action, right? So that's why it spreads in one direction more compared to the other. If the motor commands were also uncertain, it would spread in both directions. Okay, so this is, what we have done is, reproduce this famous set of experiments, geometric determinants of place fields of hippocampal neurons without invoking geometry, right? Because this was explained using this idea from geometry, but we don't need any of that here. And this is just one example. In our paper, we have some dozens of examples of things that are attributed to space and geometry, not needing any of that, just falling out of the model without that. In fact, let me show you a few things. One is this idea that, oh, in the center of a room, place fields are bigger compared to the things in the periphery. This is something that you see over and over in the literature, and that is easily explainable. It's so what will happen if we make this arena bigger? And this is also the role of grid cells will come into the picture at this point. Right now, if we make the arena bigger and bigger, what will happen is that there is less distinguishing sensations in the middle, and the learning will start faltering. It won't be able to recover a very good graph of the environment if you make the arena bigger and bigger without any additional help. And that's what happens in the model too. If you just keep making the environment bigger, the learned graphs are not as good as the one-to-one correspondence between locations that you had earlier. And when that happens, when you measure the place field of the neurons here, they will just look bigger because now they're representing more of the they're aliased. So they're, multiple locations look the same to that neuron, and that's why the place fields get bigger in the center compared to the periphery. And also you'll see that, oh, it gets elongated in this direction when you measure along the boundary, etc. But that's all because of the pattern of aliasing. Okay, here is a different experiment, completely different experiment, nothing to do with space at all. And trying to explain it using space completely fails, and sequence learning explains it very well. So in this experiment, the rat is running 4 rounds around an arena. It runs 1 lap, then another lap, another lap, another lap, and it's rewarded at the 4th lap. Something they put it at the first, but this thing repeats, so you can think of it as it gets a reward at the end of the 4th lap. And what they show in the experiments is that a different set of neurons fire for the first lap. This is after learning. A different set of neurons fire for the first lap. Another set of neurons fire for the second lap. Another set of, so it's actually counting the laps. It's not representing the location. It's representing location and the lap number. Okay, which makes sense in our model because it's just it's just learning the sequence. The sequence is ABCD, 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 reward, right? That's a sequence, and it will learn that. So if you put our model to it, it would really, this is not how, this is just a caricature representation. It gets different sensations at different locations, and the sensations are areas too. But if you just make a caricature version of it, the aliasing is just when it goes around the lap second time. So this will be the first lap, then it comes back. This will be the second lap, then it comes back. This will be the third lap, and then the reward. That's the graph it will learn. And now it makes sense why they get this curve here. You can see that when the first time that is here, this neuron will be active. When it comes back, this second clone will be active, and when it comes back again, this third clone will be active. But they will have partial activations because of probabilistic smoothing, or just think of it as noise. So that's why you actually have these partial observation, partial activation traces here. And the model has it too. So you can see that these partial observation traces here, while this is the lap counting plus location neuron. Again, just falls out of sequence learning without additional things. And they have experiments of different kinds of remapping. What if you let the rat? What if you move the reward from the end of the lap, third lap, to end of the fourth lap, which is like elongating the room. You can think of it, right? So the responses will remap, and it will say, oh, now it will start counting the fourth lap as the third lap, and the previous ones will kind of remap accordingly. So it's all of them are explainable. It all looks mysterious when you're thinking in a space-centric framework, but it all is very sensible when you think of sequential decision making. Okay, so what is the model? I explained it in terms of clones and graphs and everything, but if you are familiar with hidden Markov models, it's a very easy model to understand. It's an overcomplete hidden Markov model. The core of the model, it will generalize from that in a second. The clones that I'm talking about are all hidden states. The number of hidden states are way more than the number of observations. And so you can think of a hidden Markov model with a particular emission structure. In a hidden Markov model, all the latent states are allowed to emit any of the observations. But here we are restricting. We are basically saying these set of latent states are hardwired to emit only this observation. These other set of latent states are hardwired to only observe this other observation. So the emission matrix is hard-coded in a particular form, and the transition matrix is learned using the standard Baum-Welch algorithm. And that's we have to do a few additional tricks to get it into a graph form. So we first learn using EM, which is the soft learning, and then after learning we do. So what happens is that if you just use EM, it will learn multiple copies of this graph in the soft form, and they will be overlapping with each other. If you take that graph, the nice clean graph that I showed, and imagine having multiple copies of them weighted and superposed together. That's what EM will learn. Then we run a consolidation pass called just the hard decision version of EM. It's called the Viterbi EM, which just collapses all these things into one graph, which produces that clean graph. So it's this model plus a learning procedure that creates that graph. I'll skip this one. It's just how do you act actions to that model. Okay, generalizing from here to I started with saying you can actually use continuous observations, and that is very simple. Instead of saying, oh, we are observing color, we observe a frame of the visual input, but it passes through a vector quantizer. This can be a VQE or k-means, doesn't matter really. So in reality, basically, you have to think of what is input to the model is just numbers, and the numbers can come from color or a quantized visual sensation. And so you can let the model run in 3D rooms with aliasing, and it will learn the layout of the room. And we have ran it in quite complex 3D environments, and it works just fine in terms of learning the layout graph.
Where are all these rooms stored? In Toronto or?
So it will be a combination of hippocampus, CA1 to CA3, and the prefrontal cortex. I will come to that. Okay, and also this can be used to navigate in the environment. It's not just a model that shows place fields. No, you can actually run the model to navigate in the environment and actually do planning in the environment. I'll skip the video in the interest of time, but just imagine that the agent can actually walk, and it can see the visual. Can see what is going to come next, etc. Oops. Okay, I played it anyway. Okay. Yeah, that's just the agent walking in the room to get to the green apple, and it's a planned sequence of actions. Okay. And recently, completely independent of us, there was this paper from Genere Labs, and they have this thing saying, oh, learning produces a cognitive map in the form of orthogonalized state machine. And this orthogonalized state machine is basically CSCG, our model. And it was striking what they said in that paper. In addition, CSCG generated using learned transaminatic progress to various stages during its training that bear a striking resemblance to representations of neural activity observed in the mice, etc. And they tried many different models. They tried transformers, LSTMs, etc. But CSCG provided the best match for the things that we observed, which was quite pleasing for us, because they didn't tell us anything about it until they published until they wrote the paper. Okay, so some of the advantages of this representation: one is efficient planning. You actually have a graph, so you can actually do Dijkstra on that graph, or message passing. You can, if you want to take care of uncertainty, you do message passing on that graph. That is way more efficient than just having an RNN which predicts next states, and then you have to do stochastic sampling to plan. Doing stochastic sampling to plan is very hard if you want to do a long distance planning, because you can only just hit it randomly. In fact, we have a paper showing that exponential complexity in just the number of sequences that you have to draw. But if you have a graph, you can plan efficiently. You can also rapidly modify the graph, because it's a structure that you carry in your brain. If the environment changes, if your actions do not result in the same outcome that you expect, and it repeats, you can quickly modify the graph to reflect that environment, and it produces strong transfer and composability. So I won't explain all of these things. Let me talk about the strong transfer and composability as the next two things. So this is the idea of cognitive maps to schemas. So I'm just going to draw the graph again. So here is the arena the rat is in. Imagine that we learned the graph in this form. This graph, remember, I'm just, this is the real graph, but if you lay it out differently, this graph looks like this, and this corresponds to this arena. All these green clones are wired to the green observations, etc. You are familiar with this one, right? Okay, now imagine that I take this graph and disconnect it from the observations. This is how it is connected to observations. I just disconnect it. I still have a latent graph here, and it has some structure in it. What the structure says, all these clones are supposed to connect to an observation. We just don't know which observation it is supposed to connect to. And all these clones are supposed to connect to the same observation, different from this one. We just don't know which one it is. So now imagine that you encounter a new room which has the same geometry as this room, but just different color. The wall colors are different. So you encounter this room where the carpet is pink and the wall colors are like this, etc. What do? Do you want to learn this room from scratch the way you did before, or you want to keep this latent structure and just learn how to wire to the observations, right? Because the latent structure already has the geometry of an abstract room, which can have different paintings on the walls. So in this case, you can just learn what was the color. So you can keep this latent graph structure with slots in them and just learn the color that falls into the slots, which is just rewiring this emission matrix. So it was wired like this. It will get wired like this. So learning this rewiring is much easier than learning the latent structure. So this is the core idea of schemas. You can just rebind to learn the coloring of this graph, as opposed to learning the graph itself when you encounter environment. So here's the story. Suppose you have experience with a whole bunch of environments. What you do is you learn these environments initially just as you're learning the environment. So from this environment, you learn this latent graph, this one, this latent graph. Now you disconnect it from the specific observations. And what you have is a set of latent graphs which correspond to geometry really. And then when you encounter a new environment, this is a new environment. Well, in this case, just a familiar environment with different coloring. And instead of trying to learn that environment de novo, you just are trying to learn the coloring of them. In fact, you are trying to do all these things simultaneously. You have a set of existing schemas. When you encounter new situations, you are seeing whether it matches any of these existing schemas, while also simultaneously trying to learn something new. You don't have to learn this, so you can try to do sequence learning at the same time as doing this, and all these things can happen in parallel. And instantaneously, whichever model is giving the best likelihood, you keep using that, right? So you can learn all these emission matrices in parallel, which is very efficient. So you're trying to match all these schemas at the same time. And in this case, it will match this schema because it's the coloring, and it's extremely fast compared to learning the graph from scratch. Now, what if and this one, the environment doesn't need to be exact, and I can have size variations, etc. It still works because you saw that the animal was able to walk in an elongated environment just fine. It had some uncertainty, but overall, if you ask the animal to go to the blue boundary from the green boundary, the animal would have succeeded even in the elongated room. It just would have carried some uncertainty along the way. So here too, if the size varies, it's fine. And now, suppose you encounter a new environment which doesn't match any of the schemas, but it can be explained using a composition of schemas. You are just laying out the schemas in place to compose the environment. So you can think of a new environment which is an unknown composition of unknown number of existing schemas, and you need to learn that. So now you're learning a transition matrix between the schemas while also learning the coloring of each of those schemas. So you're simultaneously inferring the composition and emission bindings, and this also works reasonably well to learn large environments. And here is an example of something like that happening. And with schemas, it learns very fast with very few training examples, and without schemas, it takes a long time. This also explains how place fields can exist very quickly because that has prior experience and it's carrying schemas around in its head. Okay, and you can also plan through unknown environments that the agent hasn't experienced. I will skip this because it's reasonably obvious. I think if you have a schema for this environment, even if it's a new environment, you have seen only part of the environment. You have already inferred the schema, and so you can actually plan the shortcuts in that environment, even though you haven't seen this part that you're walking through. Again, the real life example is this. Suppose you walk in one floor of that one environment, and the second floor has identical layout, right? You can plan shortcuts in the second environment through novel areas that you have experienced, and you will not only be able to plan the shortcut, you will also be able to predict that I don't know this environment, I know the geometry, but I'm not going to predict the furniture that I'm going to see. Because the furniture can be different even though the geometry is the same, right? So you also know which areas are uncertain that you're going to pass through. So that's, and the model can do that too. Okay, these two things can be put together to explain this idea of in-context learning. Let me go through that very quickly. So you must all be familiar with this idea of in-context learning in transformers. So here is an example. Suppose I give this prompt as the input. XMCFG, output is CFMGX. Okay, input is this. What should be the output? So you can see that in this one, I think I just reversed it or circularly rotated it. No, I just reversed it. Okay, so the input to output mapping is reversal, and these are entirely new symbols the model hasn't experienced before. In this example, it will still be able to output reverse as the answer. This is an example of in-context learning in transformers where it works on very novel prompts. So how does our model connect to language and everything? Because it's just sequence learning. So what is aliasing in language? The same word can occur in different contexts. For example, bank can be one bank rover, or it can be a river bank resort. So bank can be a money bank or a river bank, and they will have different clones after the model learns because of the different contexts. Okay, and it can produce transitive generalization, flexible branching, etc. But so instead of sensations, visual sensations, you can feed text into the model, words into the model, and it will learn these latent graphs corresponding to latent transitions, and the different clones will have the different meanings associated with words when words have multiple meanings in different contexts. Now, in context learning can be explained as dynamically by rebinding your emission matrices on the fly. So let me just explain it using our geometry intuition. Okay, so imagine that we learned one room, which is like learning one sentence structure or one algorithm. And then we are encountering new room. We saw that we can just relearn the emission metrics. But what if only a portion of the room changes? We don't need to relearn the whole emission matrix. You can just relearn part of the emission matrix. Does that make sense? Just one wall changed in color. So, and you can keep doing this on the fly. That is, you're always assuming that your emission matrix needs to be updated, and you can keep doing this dynamically as the observations come in. The observations are different from what you expect, or you keep updating the emission matrix dynamically. And we call it, you can call it rebinding based on surprise, or rapid rebinding or anything. But it's just keeping the emission matrix to be flexible and changing very dynamically on the fly. And it's a very special case of expectation maximization. You can think of it as you are running the same algorithm, the learning algorithm, but just running it online. So you're constantly adapting it. So how does it work mechanistically? So keep in mind that the model can learn all these algorithms. For example, if you teach it to reverse a sequence, so if we give it examples of A, B, C, D, E, followed by the reverse of that sequence, it will learn A, B, C, D, E, followed by E, D, C, D, A. So it will. This is how it will be represented in the model, and you can also have a length generalized version of it where it can reverse any sequence up to length 3 to 5. But the algorithm is bound to the symbols that you trained on, right? This is a reversing circuit for ABCDE. You want it to be a reversing circuit for arbitrary symbols which are presented in context. All you do is you be able to do this symbol rebinding dynamically as the inputs come in. And that's easy for if you have only one algorithm, but in reality, you will have multiple algorithms that you've learned. So it could be, for example, you have transpose, row columns, diagonal, circular shift, transposing a matrix, reverse, all these algorithms, these are all can be learned by CSCG, so it's just sequences. They're all like different rooms in your brain. And when a new sequence comes in, which is sensations from a new room, which the sensations might look very different, but they correspond to the same geometry. Like reversing can be using very different symbols compared to what you're trained on, but it's still reversing. And so you can, when a sequence comes in, you can simultaneously infer the bindings and find out what the algorithm is. You have to do that both simultaneously. You're finding, oh, which room might I be in given this sequence, which requires doing this rebinding dynamically while inferring what latent room you are in. And surprisingly for us, this happens very fast if you just do this expectation maximization on the emission matrix. It within one iteration of the emission matrix update, it converges, and so initially it doesn't know whether it is in this room or this room or this room, but after one iteration it quickly figures out, oh, I am in this particular algorithm. Yeah, and we can see that these two look very similar. One is actually reverse, one is circular shift, and initially both of them in one forward pass, both of them are active. After one iteration, you can see that they converge onto different latent algorithms. So that is in context learning. So in context learning in our model is, you can think of it as simultaneously inferring the schema and the binding from the observation. And you can think of this fast rebinding as behavioral time scale plasticity. Okay, now in the last 5 minutes or something, I'll talk about how to connect this to a microcircuit model. So this, I have to use a little bit of history here. So in 2017 or something, we published this model, a generative vision model that breaks CAPTCHAs, and that was a purely computational model. There was inspiration from biology, but the model was explained in pure machine learning terms. And then we wrote a paper connecting it to biology, where basically saying, we are given this graphical model, how do you connect it to biology? And I'm going to do the same process for this one. What we have currently is the computational model. You have to connect to biology. I'm just going to explain the steps involved in that. Okay. So, so what if you have an algorithmic model? Here are the computations. The neuroanatomy and physiology gives you a bunch of constraints on that, how that computation can be done in biology. And of course, the properties of the world also gives you a bunch of constraints, and you're trying to triangulate between these constraints to produce a working microcircuit model. And it's kind of filling in jigsaw piece puzzles because a lot of it, because you already have a working system, you're trying to see how it might be implemented in biology. Now, all I'm going to give you is some hints on what is involved in this conversion. Remember that I am always explaining it in terms of graph computations and a graphical model. But a graphical model is not directly translated. The nodes in the graph are not one-to-one correspondent, like they are one-to-many correspondence with biology. Each node in the graph will need to be implemented by multiple neurons, and I'll explain why. So this is a simple graphical model. It's called the Noisier model, where, and what the model is saying is that, oh, when this feature is on, feature A is on, these pixels are on. And this node is saying, if this feature is on, these 3 pixels are on, C is saying these 2 pixels are on. And they combine in a OR fashion. If A and B are on, then still this one is on in a noisier manner. Okay, that's what the graphical model is saying. And this is a graphical model that can create explaining away effects. For example, if you input if the input is all 3 pixels are on, it will do a forward pass through this model where all these neurons are on, but then have a backward pass where it will turn off these 2 other neurons and only one of them will be on, like, which is a graphical model computation. But neurons can only communicate in one direction. You cannot have a neuron have a forward and a backward message in the same neuron. So if you want to implement this graphical model and these computations, you need to have multiple copies of these neurons. So if I take this node B, the computations that happen in that node are messages coming in from below, need to be multiplied together and messages need to be sent out, right? So this particular node will be implemented using these 4 neurons, and they correspond to different messages coming in and going out. Similarly, this factor here is implemented using this set of neurons. So it's a one-to-many mapping that needs to happen. So when think of all the nodes that I showed in my graphical model, those nodes involve both forward computation and backward computation. But they cannot be both in the same, the forward and backward cannot be in the same neuron. In biology, they need to be in different neurons. And that's effectively the trick in making this work. So keep in mind that an organism has to do a lot more than just forward backward computation. Suppose I am, I am an agent acting in the world. I need to maintain my state. I need to know where I am currently, or what my latent state is. I need to keep that one active. But I also want to plan forward. I want to see, I want to run different scenarios in my head. I want to be able to find out where food might be after this talk. And I want to do that thinking in my brain without disrupting my knowledge about where the current state is. If it is a computer, I can just cache it. I can just say, take the current state, put it in memory, reload it, all those things. In the brain, you can't do that, most likely, for other than in some working systems with working memory. So you need to have different neurons to represent your current state, a different set of neurons to do the planning, but they are all talking about the same thing. You just don't want the computations to mess up each other. Just like you don't want the forward computations to mess up the backward computations. You want them to be on different neurons. So the theory I am working on is that these CA3, CA1, PFC are all have different versions of the same graph for different aspects of the computation that needs to happen. So, so here is the sketch of the theory. CA3 is where the learning of the maps actually happen, where this learning this transition graphs happen. Dentate gyrus is the vector quantizer and also the emission matrices. So anything that is, people call us pattern separation. If you read hippocampus literature, they will call about pattern separation. That happens in dentate gyrus, and that's also where all the emission matrices are stored. CA3 is where the graph learning happens, and CA1 has a copy of that graph. It's an exact same graph, but it's used for planning. You have that graph so that I can plan shortcuts, and maybe I can even dynamically change the graph if the environment changes, etc. And so you can think of it, CA1 as doing the planning, also having a fast updated version of these graphs, which can then get consolidated later after an episodic experience. And prefrontal cortex is where you take these graphs and disconnect it from the observations and keep the abstract versions of them so that you can combine and recombine them. Of course, that means prefrontal cortex has to have some mechanism for communicating with dentate gyrus and gating them, etc. I haven't figured that out. I'm pretty sure the connections have to exist. One thing is definitely sure, that you need to have connections going from CA3 to dentate gyrus because that's what results in picking up the right emission matrices and doing the rebinding. So many of the models do not have a feedback connections from CA3 to dentate gyrus, but this model kind of requires it for this rebinding to happen. So I'm pretty sure there will be something from prefrontal cortex to dentate gyrus to as a prediction. Okay, so that's the sketch of the microcircuit model. I hope to, the visual microcircuit model is complete in the sense that it can reproduce all the computations of the other one. I hope to create a version of that for the hippocampus and the prefrontal cortex. We'll see where it lands. So that's the end of my talk. So here are the papers related to this one. Everything. Yeah, so everything other than this one is this is all on archive, or everything is published other than this one, which we haven't managed to publish yet.
Great talk, really exciting work. So I want to understand the components of your model better. So is it fair to say it's a autoregressive generative model?
It's not autoregressive. Okay, so the question, I am supposed to repeat the question. The question was whether the model is an autoregressive generative model. It is not autoregressive. It's a generative model, but it's not autoregressive. It's a latent state generative model. It's a latent variable generative model. So think of it. It is more similar to RNNs than to transformers. Transformers, you have to feed the input back into the, it's a fixed context window and you're sliding it. That's not how it is. This one has a infinite recurrent state. That's keeping. Yeah.
So do you ultimately learn a joint distribution between the sensory data and the weight variables?
Yeah. Yes. Yeah.
And have you done any evaluation? So in the architecture, you have this hardwired connection between like gray to gray function.
Yeah.
Have you done any evaluations in the loss function or the architecture to see like which component really contributes to that?
Yeah. So the architecture definitely. So if you do a standard hidden Markov model, it doesn't learn. It doesn't, because it gets stuck in local minimum. So you can think of it as analogous to having. It's analogous to the convolutional neural network versus a standard neural network. Convolution is an assumption that you put in, right? So here it's equivalent to a convolution. It's a convolution in the latent space. You can think of it as all these latent space neurons are hardwired to have the same emission. Just like convolution is basically saying all these neurons which are displaced in space are supposed to have the same weight matrix. So it's a different way convolution appears, but and the effects are similar. Actually, much more dramatic in the case of hidden Markov model, because hidden Markov model local minima problems are much worse compared to, especially when the model is overcomplete. The number of hidden states is way more than the number of observations. Yes.
So in your model, there's nothing special about wall.
No. Yeah.
Like in the slide you have different room sizes.
Yeah.
Is this a way to differentiate the model from the geometric view?
Yeah. I mean, yeah, there is basically nothing in the model is about geometry. So it was not a that's how the experiment is done. The experiment is with walls having different sensations from the things in the middle. We are just trying to reproduce that experiment in the right setting. So it's not explicitly to disprove it, but basically saying that this is how the setting in which the model has to work. The only sensations is the sensory observations, not the locations.
So it seems like, one of the running themes here, you keep on like you just said, you don't need geometry.
Yes.
You just do it with sequences.
Yeah.
And so I guess one interpretation of this is interesting as an existence proof that, look, just leaving that aside, just sequence information is enough alone to kind of these graphs and so forth.
Yeah.
But I guess I wonder where you're coming from this in terms of thinking about brain. Like from a normative point of view.
Yeah.
Is this good? Why is it good to dump geometry? Or is that your view? Or I mean, is there some advantage to just purely sort of thinking in terms of sequences and not geometric way? Because I'm just kind of thinking, for animal behavior, looking at 3D worlds, geometry seems like it's humongously important to our ability to survive in the environment and form representations of that and understand where we are and all this stuff. And like, if you just sort of now boil it down to sequences, it seems like, I mean, it's cool what you're showing here, but I can foresee where that's going to be potentially fragile, right? It's like a chain is as strong as a weakest link. If I sort of leave out something in the chain, now where am I? I don't sort of, So
no, it's actually the other way around. I think having a Euclidean representation is fragile and rigid. Because you have to have kind of globally register everything into, you are, there is no slippage. You're basically saying, oh, I have to take the geometry of this. Imagine modeling this environment, right? I would take the geometry of this room, I put it in the Euclidean frame, I go to the other room, and any slippage that you have is hard to correct in that, when you have this rigid, one, you have to get the model right, all the Euclidean registration right, and anytime you are out of context, you're trying to register this global frame.
is the slippage a problem? And where does that come from?
Let's say you're going from floor 1 to floor 2. Now you have to keep all the geometry in 3D. Is that represented in voxels, or is it represented as 2D planes connected to each other? If it is,
Understanding the geometry of one room relative to another.
yeah.
More global.
Yeah, so imagine modeling this building with all the floors. When you say it is Euclidean, now is it voxels in 3D, or is it a bunch of 2D planes connected to each other? If it's a bunch of 2D planes connected to each other, you're already going away from Euclidean because it's already different. Now you're saying, oh, it's Euclidean in the 2D plane, and I'm going to take something which transports me to another 2D plane. That whole combination is not Euclidean.
Okay. Yeah, I can see. Yeah, I see what you're saying.
Yeah. And it's this is especially
Maybe just it remains to be seen. Like you're talking about, well, we're not very good at connecting this floor to that floor, but I don't know. I mean, we should probe that.
I'm saying humans are good at it, but because we're not using Euclidean,
Because
we are not using Euclidean assumption. That's why we are good at it. If we try to use Euclidean assumptions, it's much more laborious because in 3D space you have to now carry around the volumes in the voxel thing. Basically saying a different representation for every 3D point in the world. Which plus, if you are in cubes, if you're running in cubes, all that is useless. Having all the 3D volume around. What you're interested in is the connectivity of the cubes, right? So
Dileep, if you think of remapping experiments.
yeah.
And what would your model predict? So if you remap between environments where you actually reuse the same schema, would you see a difference from remapping between environments where you use different schema?
Yeah, I would think so. Yeah. I'll have to run the experiment to see what.
So you would say there is kind of no. I mean, it looks. I mean, for example, the cell assembly is active place of active cells in a particular location that has now a different color would be exactly the same, right?
The cell would be exactly the same. If you're using
so whatever cells are active in the blue environment would also be active.
Oh yeah. So this depends on a few variables, right? Basically this also connects to episodic memory, right? Which is basically saying, suppose I've experienced Simon's Institute. Okay, I have learned the schema. I have learned all the rooms here, but I also want to reuse the schema, right? But I don't want to forget Simon's Institute, right? I want to keep I want to keep the bindings to Simon's Institute while also reusing the schemas elsewhere. So I would, depending upon what my consolidation level is, did I take my experience of Simon's Institute and consolidate into a permanent memory, or did I just keep it as a one-time thing? The effects can be different. So after sleep and consolidation, how it works can be very different from how it is used on the fly. Does that make sense? You can keep it two copies.
In a way, I mean, I guess there's still something to reconcile here, because if you would say there is reuse of schemata,
Yeah.
right? You would really say there is not a complete remapping.
Correct. Yes.
And I think what you see in CA3, that's a pretty good remapping.
I mean, again, it depends. So this all depends on. So I learned a particular schema. So schema. So you can think of the schema plus grounding or binding. Schema plus binding equals m1.
Maybe you want to.
Oh. It might not be that important. I can just do this. Okay. So think of it as schema plus the binding is your memory of one environment. And if you don't keep the schema and the binding together, you forget that environment. So if you you can have multiple copies of the schema, and if you're learning a new environment that you don't want to forget the previous environment, then you are actually learning on new neurons, and so then it will be completely different. Now, I don't know what are the stages in which this happens. So, when do we decide to forget one environment? When do we decide to?
Well, but if you use new neurons, you would also need to rewire the graph, right? Somehow.
They are all copies. They are organized in a way that is very easy to rewire.
Okay.
So because they are all, so you can think of it as
But that puts a lot of strain on the rewiring somehow.
on the emission reminding, not on the transition reminding. Yeah, but it's a
So I mean, for the cells, for the transition. So you're saying you don't need rewiring.
yeah, for the
But then
transition.
it's the same cells that represent the transition.
So you can think of it. This is one simplified way of doing it. I mentioned that when we actually learn the thing, you are actually learning multiple copies of this graph superposed with each other, right? And we are cleaning it up to show this. We are cleaning it up and showing this one graph. Imagine that you actually had multiple copies of this graph learned. It doesn't change anything. It's exactly how it learns. Really, it doesn't change the behavior. It doesn't change anything. All you have to do is now, if you say, oh, these multiple copies, I'm going to start separating them into different memories. I'm going to say, oh, these one copy I'm going to rebind to one emission matrix. This other one I'm going to rebind to another emission matrix. So all the transition matrix wiring remains exactly like before. You are just rewiring them to, you're taking one and giving them one coloring, other one giving a different coloring, which is all emission matrix rewiring, which should be.
Like the transition wiring, you basically map to CA3. It puts a lot of strain on CA3. To keep all these different graphs even for one and the same environment. I don't know.
I mean, it's a in terms of organization, it's CA3 won't even know that these are two different graphs, or yeah.
I think that maybe we'll stop here, but we have, if Dileep has time, we can all go to the second floor and do a more informal discussion and continue. But if
Yeah,
you guys
sure.
have time,
Perfect.
Thank
Thank you.